I'll introduce our final speaker now, David Plew, who'll be presenting on modelling transport of invasive organisms in lakes.
We're back on time. Good afternoon. David Plew from the Institute formerly known as NIWA, now Earth Sciences New Zealand. No doubt I'm going to call it NIWA several times through this. I'm still getting used to it myself. My talk is about modelling transport of invasive organisms in lakes, and what I'll go through this afternoon is what could this modelling be used for, and give two case studies examples. One is looking at transport of Lagarosiphon in Lake Wanaka, a test case in Lake Akatano, which we use as a test to see how quickly can we set up a model that could be used as maybe a response tool, and then look at where can we go in the future with our modelling. So when I'm talking about lake models, I'm talking about lake physical models, or physics models rather. So computer simulations that simulate the physical processes, the water currents, the water movement, the temperature, how a lake stratifies in summer, how it gets mixed up by the wind. And these can be used to predict where organisms are transported to, or possibly where they've come from. Other uses, they can give information about the habitat suitability, things such as the water speed, the water temperature, the amount of light. This could be overlaid with other information such as depth or substrate type to work out what's the risk of fragments moving from one area to another, or an organism from one area to another. And lake hydrodynamic models can be coupled with water quality models to predict things such as oxygen, nutrient, productivity, meaning algal growth. Where can they be useful? If you consider, in this case, Lagarosiphon in its what's called here the invasion cycle, where fragments break off, they're transported, then they establish and grow and take over, and then eventually more fragments break off. Where the hydrodynamic models are most likely to be useful is in that transport phase when fragments have broken off, they're being moved by the currents to where they re-establish. And for other species that also have a transport phase, such as our golden clam here, in part of their life cycle where there's a larvae that's transported around by the currents. So again, that's the area where these hydrodynamics are most likely to be useful. In terms of Lake Wanaka, in the Lake Wanaka Lagarosiphon management plan, there's a goal that Lagarosiphon is progressively contained and eradication gains are maintained. If you were here earlier when Jordan gave his talk, you would have seen some of the progress being made in that space already. One of the objectives in here is that the source weed beds of drift into the eradication zone are controlled. I'll come back to that in a moment. And one of the milestones is that the transport patterns for weed fragments are modelled. And some of the work I'm presenting here goes towards that. Here's Lake Wanaka split into two. There's a massive earthquake, broke the lake into two. Most of the lake, the northern part of the lake, is what's currently called the eradication zone. So I'm talking here about lots of knowledge, the work that Linz and Boffa-Miskell have been doing to eradicate it, and these plots we've borrowed from them. So north of this containment line, which stretches from roughly Bishop's Bay across to the peninsula here, Lagarosiphon has been largely eradicated. There's ongoing monitoring, and individual plants are removed by hand weeding by divers. As Jordan mentioned, there's the Western Target Control Zone, where a lot of recent efforts have been used using Hessian barriers on the lake bed, suction dredging, spraying, even hand removal, to largely eliminate Lagarosiphon from the western part of the zone of the lake. And now the intention is to move the buffer zone or the containment line further into Roy's Bay, and hopefully eventually remove Lagarosiphon from Roy's Bay and then Stevenson's Inlet as well. So I'm going to present some modelling here where we have simulated how fragments of Lagarosiphon that might be broken off will be transported around the lake. In the model here, we're releasing plant fragments from 6 locations, 3 of those in Roy's Bay, and 2 in Stevenson's Inlet and 1 just outside Stevenson's Inlet. When this starts playing in a moment, you'll see little dots moving around, which are plant fragments or particles representing plant fragments. They're released every hour. they're given a lifespan of 14 days. The reason we give them a lifespan is that over time their viability will decrease, and 14 days is a bit of a punt about how long they'll last. And also we're assuming that particles that drift below 10 metres depth, there's evidence suggesting that at pressure below 10 metres, the pressure is enough to cause these fragments to degas, and then they'll sink and be lost. So here we go. Exciting, isn't it? Probably the things just to note are particles that are chemical fragments released up in Stevenson's Inlet tend to stay in that part of the lake. Down in Waterfall Creek or Stony Creek in Roy's Bay, these fragments don't move very far either. They tend to stay quite local. But every so often you'll see fragments from near the Yacht Club or even Quartz Creek come around the peninsula and head up into the northern part of the lake. This happens when there's a strong southerly blowing, so the wind is the major driver of water currents in the lake and of the transport. These particles get fainter over time. That's indicating their age, so a darkly coloured particle is a fresh one, a faint one is an older particle. Now, I'm sure we could watch this for ages. Luckily I've only got a month here rather than a whole year. I mentioned briefly that particles below 10 metres depth we've removed. The properties that we give our particles to represent the plant fragments make a difference on how they are transported in our model. For example, if we say these are buoyant particles and they stay in the upper 10 metres, that's where the currents are strongest, and so these fragments will be transported the furthest. So fragments released from up in the top of Stevenson's Inlet, a few of them over a period of a simulation of a year, some of them do make it out of Stevenson's Inlet. Now if we give no limit on their depth, we allow these fragments to get carried below 10 metres, they'll still stay viable in our model, they'll just get transported around. Few of them make them out of the inlet because in this case there's a sill here at the entrance to Stevenson's Inlet with a depth of about 10 metres, and a lot of the deep water actually stays trapped inside there. And our final case, this is the one we're going with. Those fragments, they get pushed below 10 metres, we're assuming again they're degassed, they sink, they're lost, they go to depths that they won't survive. We think this was probably the most realistic for lagrocycling. Some results. I'm going to show two types of results. The first set of results are looking at what's the probability a particle or fragment released from one of these locations can be detected somewhere in the lake. So in our model has a resolution of a 100 metre grid cell, and I've calculated here the number of fragments that have passed through each of those grid cells, and they're coloured here. So it's a log scale, so red being there's a very high probability that a particle will pass through that cell, dark blue being very low. So for particles fragments released from the top of Stevenson's Inlet, again, most of them will stay very close to where they're released from. A few travel out into the outer part, but quite low probability. Coming across to Quartz Creek is the other extreme. These are released just outside Stevenson's Inlet. Again, most of them stay down in this part of the lake. A fair few head out towards the outlet. A small number indicated by this sort of pale blue, so maybe around 3% will make it out to this part of the lake. Very few do make it right up into the northern part of the lake, going quite a big walkabout. These are individual particles. As an indication, there's about close to 10,000 particles released during the simulation, so only one or two of them have made it that far up the lake. These are the ones from Stevenson's Inlet, and looking at the ones released in Roy's Bay, as you remember from the little dots moving around, the ones released from Waterfall Creek don't tend to go very far. There's one or two that do get pushed up past the Matukituki. Stony Creek tend to go a little bit further, but again, the vast majority stay within Roy's Bay. But the ones released near the Yacht Club, a lot of them do travel a lot further. Again, while most of them stay within the larger Roy's Bay area, some do travel right up, even a few right into the top part of the lake. Again, most of that north motion happens in a strong southerly storm. The other question besides what's a probability that a fragment arrives in a location is how long does it take to get there? And particularly what's the shortest amount of time it takes a fragment to get there, thinking that those ones that have travelled there fastest are most likely to be viable. And the plot here shows, again looking at any one of these locations, the colour indicates what's the fastest a plant fragment will have arrived from one of these source locations. So let's pick on the yacht club, for example. So the ones that have made it up onto the top end of the lake have taken to maybe sort of 7 to 10 days to travel that far in the lake. Other things to note is that perhaps going back, only a few fragments make their way into the Bishops Bay area. None really get through to Glendew Bay or Paddock Bay that are released from any of these locations here. As a bit of a reality check how the model was doing, this is data collected from the eradication zone. So these are surveys done every 6 to 24 months where divers are looking for individual plants and then hand weeding them, collecting them. And the percentages are the proportion of the total number of plants captured from that zone and where they've come from. Don't get too hung up on the numbers because the length of each of these zones is quite different. For example, the West Wanaka zone is very small compared to the Bishops Bay one. But the main message is most of the fragments that are finding are along the Bishops Bay coast and along the western side, not so much along the eastern side of the lake. And that's broadly consistent with what the particle tracking model is suggesting, and that the particles that do travel up here are generally heading into this part along the western side, and not so many of them are travelling up the eastern side. So just the conclusions from our modelling for Lake Wanaka. The modelling shows that Stevenson's Inlet is quite isolated from the rest of the lake, so fragments that are released in there tend to not make it out, and only few that are released outside there make it in there. The western shores are the most exposed to fragments released from this part of the lake, which is consistent with what the modelling shows. And of the 6 locations we chose, the Yacht Club and Quartz Creek were the most dispersive sites, are the ones that release the particles that travel the furthest. And those are the regions that may be, I guess, the greatest risk of being a source for the rest of the lake. With Wanaka, we've been working on that lake for at NIWA, formerly NIWA, now West New Zealand. We've been working there for 3 years, developing our model, which is a fantastic luxury, but if it takes 3 years to develop a model, it's not probably going to be useful in many locations. So we set up a project inside Earth Sciences New Zealand, formerly NIWA, to see how quickly can we get a lake model set up where we don't already have one. What are the roadblocks to doing that quickly? And what can we do to streamline this process in the future? And so we gave ourselves a time allocation of about a week to see where we could get to. We chose Lake Okataina as a test case for a couple of reasons. One, there's other projects going on there. What I'll show you shortly is simulations where we've released particles that could resemble any invasive organism that's transported passively by currents from the boat ramp. I was thinking the boat ramp is probably the most likely place where something might come into the lake. So particles, dots are released from the boat ramp. This is a 3-dimensional numerical model. The model has a resolution of 50 metres. Those fragments are released every hour for a year. This time I've just given a 7-day lifespan, but it's modelling, we can do what we like. So again, the main message is most of the particles tend to stay in close to where they're released from, but every so often a few of them go on a big walkabout down to the southern part of the lake, and that's again driven by weather conditions. This model is driven by the weather observations from the Rotorua Airport, being the closest weather station I could find. The plots here show how far particles have moved over a certain time frame. So within the first 6 hours of being released, this is again aggregated over the whole year, but within 6 hours, this is how far our particles released. So mostly staying quite close to the boat ramp. Over 24 hours, and then after 5 days, where a few have made it down to the southern part of the lake. And if you run this for longer, for 2 weeks, you'll find that probably every part of the lake, there's a chance that fragments could travel there. Showing those same probability plots. What's the probability that any fragment passes through the zone? Again, the probability is most highest close to where fragments are released from, as you'd expect. Very low probability that within 7 days any fragment has found its way, or particle that's found its way down to the southern part of the lake. And how quickly do they travel? So to reach, say, the few that have made it down this part of the lake have taken around sort of 5 to 6 days. Within 1 to 2 days, particles will travel down to cover most of the top third of the lake. So what's next? What limitations or gaps have we discovered trying to set up models quickly, and what can we do to address this? The first one is data availability. To set up a hydrodynamic model of this sort, we need bathymetry data. And we have bathymetry data in digital format for about 50 lakes. And there are lake charts, which many have been scanned but not digitized, so for another 120 lakes perhaps. We do need local weather. Often weather observations are not close to the lake, or can be not have all the variables that we need. And there's often gaps in those data which require some filling. And inflows and outflows. Like Akatana is a good one to pick on because it has no natural outflow, so it's mostly the wind that pushes things around. But if you're looking at lakes in the Waikato River system, for example, then river flows will be quite an important driver for transport. And behaviours, at the moment we're just treating anything as a passive object that gets transported only by currents, and we're not including things such as their buoyancy, and we don't really understand the lifespan, and we haven't looked at and tried to include any kind of active motion. Ways we can improve this. There are weather models that cover all of New Zealand at high resolution. The institute formerly known as NIWA has these models inside, so we're looking at ways to tap into those models and do this in a more automated way. Similarly for river flows, not every river flow is measured, but there is a model for every river flow. It's not calibrated, so do you trust it? And the more we do this, the more we can automate our workflow. I'd strongly encourage any behaviours to understand the properties of the thing that you're trying to track, whether it's its buoyancy, its lifespan. And ultimately we can do more sophisticated things like backwards tracking. So you found an organism, where does it come from? Let's run the things backwards. And there's even the potential to run short-term forecasts of, say, 2 to 7 days. Just like to say thank you, acknowledge again the work of Linz and Boffamiskell, and thank you for sharing your data, even if you weren't quite aware that I was going to present it. The colleagues from the Institute, formerly known as NIWA, for their feedback, and the MBE for funding this work through 2 of our flagship programs. Is your question, can we go to afternoon tea now?
I like afternoon tea as well. But that's really fascinating stuff, David. A great model, and like you say, a lot of what you're seeing in the model is what we're observing in Lake Wanaka from the data we're collecting. I'm wondering about your 6 points that you chose. How many more points can you add, and can we space them 200 metres around all those weed beds? Is that possible, or is you talking about computer power that's too demanding?
It's certainly possible. Yeah, there's lots of tricks we can play with the number of particles. Just to give you a sense, that Wanaka model that I'm showing there, it takes about 3 to 4 days to run on a reasonably grunty desktop machine. The Akatana one takes about 6 hours. So the size of the lake matters, the number of particles matters, but we can do it. We can run several models at the same time and have some particles in one location, some particles in another location. So yes, it can be done.
I love a heat map of the most high risk weed beds that are contributing to that fragmentation further up the lake.
Yeah.
Yeah, David. Yeah, thanks. Very cool. Thank you. The Okatoma model, is interesting. But what we've seen around more water in that lake, obviously, it was probably more related to releases from anchors or other things. So, is it possible to incorporate in key fishing spots that are known?
Yes, we can. It's a model. We can release particles anywhere.
Right.
Yeah. The question is knowing where to start from. Yeah.
Right, yeah. So you're thinking about a lot of those like key fishing spots would already, they would be known locations.
Yes.
Great, thank you very much, David. And thank you to all of our speakers. And now it's time for afternoon teas, but we'll put our hands together for